Good afternoon, and thank you for the opportunity to present the first emergency neuroprotective treatment for neurotrauma. So I'm going to present you an alternative approach to treating permanent disability caused by spinal cord injury. Instead, to repair the damage, to prevent it. But how we can prevent the injury? We can prevent an accident, but we can prevent the damage caused by accident. And I'm talking about emergency treatment that will be administered in the ambulances and trauma units. Today, there is no emergency treatment for non-types of neurotrauma. I am a neuropharmacologist with expertise in drug development. During the last 15 years, I have been working on the new emergency treatment for all types of neurotrauma. During these years, we have developed a highly effective neuroprotective treatment for neurotrauma based on biological molecules and demonstrated the effectiveness in the 2 spinal cord injury models and 2 traumatic brain injury models. So in 223, the NeuroHagana was established. And today we are in the stage of fundraising to complete our preclinical development. How does our treatment prevent the permanent disabilities? The mechanical damage, primary mechanical damage as a result of an accident, it's only 20% of the total damage. The rest is a secondary injury, which begins immediately after the primary one and responsible for the permanent disabilities after that. So our treatment prevents a secondary damage, saving up to 80% of neuronal tissue. But does it have to be an emergency treatment? The answer is yes, because timing is everything in neurotrauma. You can see here on this slide the summary of the main irreversible damage from the moment of the injury up to the years after. And you can see that the main damage, irreversible damage, happens in the first few hours. And it starts with increasing a glutamate concentration in the spinal tissue. Glutamate, the main excitatory transmitter in the central nervous system, but at high concentration is very toxic to neuronal cells, leading to irreversible neuronal damage and extensive secondary damage. So our treatment reduces glutamate concentration in the neuronal tissue, saving the tissue and prevents the secondary damage. It is a unique treatment, but not only because it's the first emergency neuroprotective treatment, but also because our treatment doesn't need to be delivered to the injury site. It works in the blood. I would try to make it shorter, the scientific part. So under normal physiological condition, the glutamate concentration in the central nervous system is very low, while at the same time in the blood, it's relatively high. Immediately after the injury, the glutamate concentration increases up to the toxic level, killing neuronal cells. It was discovered that reduction in blood glutamate concentration shortly after the injury, boosting the efflux of excess glutamate out of the central nervous system, balancing his level and preventing the secondary damage. Our treatment reduces glutamate concentration in the blood quickly and efficiently and can be administered by any team in the field. So we did a proof of concept in 2 animal models, mice, compression and contusion, moderate severe spinal cord injury. And using the transgenic mice where we can see the neuronal tissue, axons and neurons can be easily visible and follow up under a microscope. Here you can see the representative images of the injury side. Sorry. Yeah, okay. So on the left side, it's intact tissue for comparison. Then you can see 5 days after the severe compression spinal cord injury, the tissue of untreated mice. Then the center of the lesion site only after 1 injection of our treatment that was administered 4 hours after the injury. And this is the 5 injection. So you remember the previous illustration, 20% of primary injury, 80% of the secondary injury. This is what you see here. This is only a primary injury. We prevented 80% of the injury by 5 injections started 4 hours after the injury. And not only we save the tissue, they are functionable. So one of the way to see how functional, how healthy axons, it's to stain with myelin. And again, here on the left side, this is how it looks, a healthy tissue. Then untreated mice, you see no neurons survived, no axons crossing the lesion site, and destroyed myelin around. And this is the treated mice with the 5 injection. By the way, to compare the amount of axons that survived at the lesion site, it's about 80%. And a healthy myelin, so they're also functional. I'm going to present, so as it was said before, and I agree 100%, it doesn't matter statistical results at the end, the starts, but we should see the functional recovery. This is what is important to the patients. So I'm going to show you 4 videos of injured mice, 2 untreated, 2 treated, taken 48 hours after the injury and 5 weeks after the injury. I apologize to the people in the audience who are sensitive for the images of the injured animals. Let's see here. Injured mice, completely paralyzed, and 2 legs, 48 hours after the injury. And this is after 2 injections. Pretty impressive functional recovery only after 48 hours, supporting our hypothesis that we are preventing the secondary damage. This is not a regeneration, it's a prevention. 5 weeks, completely paralyzed mice, most of them completely paralyzed. Some of them recover by 10 to 15% mostly. And this is the treated mouse. So if you are not expert in the preclinical experiments, you will not see any difference between these mice and injured mice. But this mouse was also totally paralyzed the first day. And it is a summary of a motor score where you can see that our mice that were treated recover up to 80%, all of them. So when I was asked, so what is the percentage of recovery? All of them. All the mice that we done up to now, I should say about 400, all of them recovered. And all of them recovered impressively. About anti-glutamatergic treatment, you probably remember the past failures. So the glutamatic cytotoxicity phenomenon known as a main player in the neurotrauma for many, many years. And a lot of companies try to develop treatment against glutamatic cytotoxicity. Unfortunately, all of them failed in clinical trials, partially due to severe side effects, mainly of the central nervous system, and partially due to the lack of efficacy. So why we will succeed where the other failed? First of all, as I mentioned before, our treatment doesn't need to be delivered to the central nervous system, and it's based on the biological molecules, so it's safe. No systemic side effects, but not only that, no side effects in the central nervous system because the treatment only balancing the glutamate up to the normal level without interfere with any brain activities. So no central nervous system side effects. And what is the most important, I mentioned it's an emergency treatment. So it can be administered in the field to anyone with an accident without make a previous assessment of the type of neurotrauma or the severity of the damage, or if it's a neurotrauma at all, because it's safe. You know, it reminds me when I was working in intensive care units, cardiac care units, and we were instructed our patients to take aspirin 325 in case of chest pain. Because if it is a heart attack, it may reopen the cardiac vessels, but if not, no harm, no side effects. The same approach. It can save life, it can save people from disabilities, but if it's not neurotrauma, nothing happens. And the last one, when it's given shortly after the injury, it blocks a multiple signaling pathway, not only one, but most of them, and this is how we reach the 80% of the recovery. So let's stop talking about science, because I can talk about it days and weeks probably, and let's talk about business. So the numbers are big. Our company is going to be focused on 3 main types of neurotrauma: spinal cord injury, traumatic brain injury, and ischemic stroke. And only in Europe and US altogether bring us to 7.2 million cases per year, and the numbers are growing, unfortunately. I was able to reach this important point to make progress in my research thanks to the supporter, and I would like to thank Wings for Life Foundation and United States Department of Defense for funding my research. And speaking about our future plans, we had a meeting with the EMA experts where our successful completion of the proof of concept was confirmed and our future development plans were approved. And speaking about sponsors, one of the potential sponsors is the European Innovation Council, their new program for early stage startup with disruptive technologies, where we just passed the first step of selection. So we are planning to complete the preclinical development in 3 years maximum. I believe we can do it even faster, and reach 3 main milestones: to produce a primary formula, to show that the primary formula is effective as our lab formula, and to complete the toxicology study. After selection, the serious companies, each milestone was designed following their regulatory rules, and the budget was built based on their offers. So in sum, it will cost us $4.1 million to complete the preclinical development and to move on to the clinical trials. Since we completed the proof of concept, the company doesn't need to open the research lab. Therefore, we are keeping a light organization with a small team focusing on management and the supervision of outsourcing companies, and of course we will grow with the needs. And speaking about the connection with the clinicians, I do believe that it's very important. I do believe that it's important in the very early stage. So I started my career as an intensive care units nurse. IVP R&D Yona, she is a physiotherapist. She is my assistant in a study, and she is still working as a physiotherapist on the top of the research, sometimes instead of the research, unfortunately. But she's taking care of the spinal cord injury patients also today, doing a great job. And in a summary, I hope that you will join us in our mission to bring the first emergency neuroprotective treatment to the market, to everybody who needs it, and as soon as possible. Thank you.
Great, thank you so much for that presentation. It seems like a very innovative approach you're taking to treating at the point of injury. My first question was around glutamate levels and how we think about potentially dropping those beyond the normal level. Is there any risk to your therapy being too effective and having excessive drops in glutamate in these patients?
Yeah, no, we are not. We are balancing close to the normal level. It's impossible to reduce under the normal level. It's 1 micromolar, from 1 to 5. So we are reducing around up to 10, and not more than that. So no, we can't. It can't do any damage. And speaking about I continue your question, what about the blood? We're reducing it in the blood. So in the blood, first of all, the fluctuation during the day can be up to 50%, and it depends what did we eat, if it's monosodium glutamate, yes or no, if we had some physical activities, if we are excited or no. So no harms, no to peripheral system and no to the central nervous system.
Got it, great. And then I also wanted to ask about the operationalization of your therapy. How do you envision it being used in practice in the field? Is this a,
in
a
practice,
shot? Or
yes,
how
in practice, I think it will be a pre-filled syringe that we are giving it to any first aid team. Ambulances, the people who are traveling and have with them paramedics. Even to the, I would go even forward to say that at a certain stage it will be given also to the general population. If you have an accident, because our treatment can be injected also intramuscular. So if someone was involved in an accident, he can just inject it immediately.
And I assume it's very stable then at room temperature and things of that nature?
Yes, we can reach it, yes,
okay.
yes, yes.
So effectively like an EpiPen or something of that nature.
Exactly,
like an
EpiPen. Yeah, just EpiPen, it's about 15% of anaphylactic shock in the US population. Yeah. Here, you don't know who is going to be involved, unfortunately. So generally, everybody needs to have it.
Great, thank you so much for the presentation.
Thank you.